Good morning. 11 o'clock, first Sunday of the month means it's time for Chai and Why at Prithvi Theatre. Good morning to the online audience as well. Don't go far away because that's the mic.
Oh, yeah. Oh, yeah, okay.
So
I can keep it close to you.
Yeah, no, that's your phone. So it's okay. The mic is somewhere there. It doesn't matter. It's fine.
All right.
If you can't hear us, online audience, please put it in the chat. Otherwise, we won't know what's going on. It's a pleasure to welcome you all here. For those who are joining us for the first time, Chai and Why happens on the first Sunday of every month here at Prithvi Theatre, the third Sunday of every month at Ruparel College in Matunga, and if a month has a fifth Sunday, we are at somewhere live from a lab in TIFR. And just to tell you that the next 2 Sundays from now at Ruparel, the topic will be something on chemistry. We'll give you details of that at the end. Today, it's a pleasure to welcome Sundar Naganathan. Sundar is one of our very young recruits. It's just 7 months that he's joined TIFR, but he works on something so exciting that we thought this would be an awesome topic to have a Chai and Why on. Sundar is from Madurai. He started doing a BTech in biotechnology from somewhere in Chennai. Then he went to the Max Planck Institute for Cell Biology and Genetics in Dresden, Germany. Did a PhD from there, went to London as a postdoc, then to EPFL Lausanne as a postdoc, and eventually we were lucky to have grabbed him and brought him back to TIFR over here, where he works on left and right. So a very intriguing topic. I don't know what it is about, so we'll share screen and then you can go ahead. So just let me share the screen. And yeah, please put your questions and comments in the chat.
Thank you. Thank you. Next slide. Next slide.
Is that your first slide?
Yeah, that's my first slide. Just a minute.
Yeah. Yep, nice and right.
All right.
And just see if this works. Yes, it does.
Yeah, right.
Okay, and you have a pointer as well.
Yeah.
The
Cool.
pointer works only for the audience here.
Well, yeah, true. Yeah, I'll try and use the cursor here. Yeah.
Anyway, online audience, you're not getting chai.
All right. Thank you so much, Arnab, for the introduction, and thanks to Chai and Why for inviting me to discuss with you all. I won't call this as a lecture, so it's going to be a discussion. So feel free to interrupt me at any point of time if something is unclear. I can see that people of all ages are here, so I'll try and keep it as simple as possible. And just feel free to interrupt me, no hesitation. So it's purely a discussion. Okay. So I joined TIFR 7 to 8 months ago, and my lab works on. Yeah, just a minute.
It's not there. Just a second. There's a phase lag, so just waiting to the screen comes up here. Okay.
Yeah.
Okay. So now we have your thing out there, back here.
All right.
Sorry about that.
No problem. Careful. All right. So my lab works on how the left and right sides of the body are differentially specified during embryonic development. Okay. So before I go into what is left and what is right and how different organs are specified, in different parts of the body. Let me give you an overall view of how our body is organized into a three-dimensional axis. Okay, so let's say that we come across a human, or let's say that you come across a dog. Okay, so if I ask you to naively describe that animal, what would you say? You would probably say this animal looks a bit elongated. It walks on four legs. It has a couple of eyes, mouth, and probably it has a heart, and so on and so forth, right? So what if I say that, in fact, we can say the same thing with a simple sentence, which is basically our body and organs are organized along a 3D axis. Okay. So what I mean by that will become clear in a couple of slides or 2 or 3 slides down the line. Just bear with me. Okay. Now, if you look at this cartoon, you see, the skeleton and a bunch of, you don't see the organs, but you have a bunch of organs in the body, right? So you have eyes, you have the heart, you have liver, kidneys, and so on and so forth, right? So these organs are not organized randomly, right? So they have a specific position and orientation along the body axis, okay? Now, how does this all come about during embryonic development? And what is the point of having different organs in different parts of the body? That will be the topic of our discussion today. Okay. Now let me try and deconstruct little by little, sorry, how the different organs are positioned in different parts of the body. Okay. So the three-dimensional axis I have drawn here, I'll come back to this a bit later. All right. So here are two. Okay. So here are two videos. And one is a cheetah, which is running towards its prey, and another is just a snake, which is just moving towards something, right? So let's look at their sensory organs. Okay, so their sensory organs are basically positioned somewhere close to the front of the animal, right? So sensory organs, by sensory organs, I mean you can think of ears for hearing, eyes, mouth for tasting, nose for smelling, whiskers for detecting different stimuli, and of course the brain. I include the brain in sensory organs because all the processing is done in the brain for all these sensory systems, right? Now what you will see here is that all these sensory organs are arranged or positioned in the direction of movement of the animal, right? They're not randomly positioned. So they're really positioned in the direction of movement of the animal. Okay, this makes total sense, and this is called the anterior part of the animal. Anterior literally means the front part. Okay, the front part of the animal. Okay, so this makes total sense because as an animal is moving forward, it's trying to look out for a predator, or it's trying to find a prey, or it's trying to look for a mate, and so on and so forth. It really helps if all your sensory organs are in the direction of movement of the animal. Okay, so that is really the anterior part of the animal. Okay, so this is true for most of the animals that live in this world. You can think of any animal that live in this world. About 98% of all animals that live in this world have this kind of organization. Okay, of course, this is not completely true for us humans. Okay, and we have all our sensory organs similar to where you will find in a cheetah or a snake, right? So the eyes and the relative position of the eyes, ears, nose, mouth, everything is very similar across all these animals. But of course, we don't call this as the direction of movement of the animal, right? So we are moving in this manner, but the sensory organs are all positioned above the body, right? So the reason for why there is this distinction between most of the animals that live in this world and us humans is because of this. Okay, so at some point we just decided to not use two of the appendages and just walk straight. Okay, so naturally you are going to change the position of the sensory organ in the direction of movement to somewhere perpendicular to the direction of movement. Okay, so is this clear to everyone? Okay, so just because now if an animal is moving this way, now I'm just straightening the back. Now, even though you're walking in this way, the sensory organs are not positioned directly in the direction of movement. Okay, and this is purely a consequence of how we decided to just forego two of our appendages and walk with a straighter back. Okay, now obviously this had multiple advantages because None of the other animals had this advantage where you could do something at the same time as you're moving, right? Because you have freed out two of your appendages, which is your hands. Now, while you're walking, while you're moving, you can actually use these two hands for doing something else, right? Which is not possible for any of the four-legged animals, right? Right.
Yeah, people can text on their phone while walking and fall down.
Yeah, exactly. That is true. So there are some disadvantages as well, right? All right. So it provided a huge amount of advantage. For example, if I have an animal which is four-legged, and if I just straighten the back for the same height of the animal, I have a better line of sight, right? Better viewpoint, right? Otherwise, I will have a little lesser viewpoint, right? So it provided, so you can detect prey or predators much earlier than a four-legged animal of the same height. So that provided an evolutionary advantage, because now it increases your chances of survival as well as reproduction, because you are going to find a mate much earlier than someone who is on four legs, right? And the other advantages, as I mentioned, you could do something with your hands, like holding on to some tools and defend yourself while you are moving, which is again not possible for a four-legged animal. Okay. Now it is really not black and white, as I said. Now, if you have some advantages of straightening your back and walking with two legs instead of four, you have other disadvantages. Okay, and what are those disadvantages? So there are a few of them, but I'll just mention two. Okay, one very big disadvantage is that just because you have straightened your back, so here in an ancestral animal, you will have the back which is more tilted or even flatter. Now you have basically straightened your back. Now what this means is that the entire body weight is going to fall on the pelvic region, right? So which is not true for any of the four-legged animals, right? So this puts a large amount of pressure on what is called as the pelvic canal. So this is the pelvic canal, and which is also called the birth canal, right? And this had to change, its structure had to change over time in order to accommodate the increased load that you're going to place on the lower part of your body, just because you decided to walk straight. Okay. Now, one of the biggest implications of this is that this whole pelvic canal, so this is in the case of a chimpanzee, and this is in the case of humans, you can see that the pelvic canal has become more rounder and smaller in size, right? Now, this is a problem because you can only, if someone is having a baby, right? So the baby can only be for a certain amount of time within the mother because of the change in size of the pelvic canal. Okay, so because of which you will have physically undeveloped babies being delivered compared to any other animal, right? You can take any animal, right? So you can take a deer or a bird giving birth to its babies, and you would see that within hours or even immediately after birth, these babies are going to walk, swim, fly, everything, right? But this is so much in contrast to a human baby, right? Human babies take much longer, so they need like 1 year, 2 years to actually start doing any kind of physical activity. That is mainly because of we decided to walk straight. That reduced the size of the pelvic canal, so we can only accommodate babies of a certain size and certain development in our bodies before which we have to get them out, right? So that there are huge implications, because it puts a lot of social pressure on parents, especially mothers, and this means that you have to spend 1 year or 2 years taking care of the sort of relatively underdeveloped baby compared to four-legged animals. Okay, the other disadvantage that came out because of this transition is because, as I told you, the entire body weight is now falling on the lower back, right? Now, this caused, even though the structure of the pelvic canal changed, this caused a huge amount of problems with respect to lower back pain. Okay, so none of the four-legged animals have any kind of, as far as we know, have any kind of lower back pain or back-related problems. Mainly because if you look at a four-legged animal, so the back is more in an arch shape. An arch shape is a very stable structure for any kind of situation. Okay, for running, for climbing, and so on. Okay, And this is completely lost in a two-legged animal like us. And all the entire body weight, especially the head and the whole body weight, is going to fall on the back. And that over time causes a lot of backbone related disorders. Okay. So a lot of things, so you will see, if you look carefully into your own body, a lot of things that you see, a lot of disorders, a lot of pain that happens over time is actually a direct consequence of how we evolved from pre-existing species. Okay. So I'll give you one more example, which will make it even more clear. Okay. So I want to make 2 points here. One is evolution is always a trade-off. Okay. You will get some advantages by walking straight from a 4-legged animal to a 2-legged animal, but at the same time you have other disadvantages, as I just mentioned, right? The other thing that I want to mention here is that newer forms always emerge from pre-existing forms. Okay, it's not that we came out of the blue, right? So someone came and implanted us in this earth or anything like that, right? So we evolved from forms which already existed, and those forms evolved from previous existing forms, and so on and so forth. So evolution always works in a way that already existing forms are modified or fine-tuned, and newer forms and shapes emerge in the body. Okay, and this is very, very clear in the case of, for example, evolution of our limbs. Okay, and these limbs are evolutionary conserved to the fins that you see in the fish. Okay, in fact, if you carefully draw the kind of fin structure over through evolution from very primitive fishes to advanced fishes to amphibians to so on and so forth until humans, you would see a very neat transition of how fin structure evolves into a limb structure. Okay, so this is what I mean by newer forms always emerge by modifying pre-existing shapes and forms. Okay, now even though this seems very, very simple in cartoon, right? So you can just draw this in like a few hours, right? So in fact, this transition takes millions and millions of years. Okay, even this transition going from a 4-legged animal to a 2-legged animal took about a few million years to happen. Okay, because this required a large number of structures in your body to accommodate the change in posture, right? And one other posture, which is one other structure which basically had to change significantly, is the back of your neck. There is a little hole in the back of your neck where the brain is connected to your spinal cord, which runs below. And this back of the neck, where the hole is called the foramen. Okay, so this foramen is usually tilted in in 4-legged animals, because the spinal cord is tilted and the brain is like that. So the foramen hole is sort of tilted in this way. Now, as the back became straighter, this foramen had to also change its position and it became bigger over time. And that is another transition which required millions of years to happen. Okay, so the point that I wanted to make here was that you always have to think about where you evolved from before you consider what kind of advantages and disadvantages as a species you have. Okay, yeah, yeah, yeah.
On planet Earth, is it because of the composition there was more water 20 million years ago, and then the water reduced and then had land, so as a result of which transition happened, and the animal had to leave because of less water?
I don't
repeat the question for the online audience.
All right. So the question is whether the transition from fins to limbs happened because of the change in the amount of water in the world over time. Okay. So I don't know. I don't know how much the amount of water changed over time, but at some point in evolution, animals decided to move more into land. Okay. So it could be a direct consequence of having slightly lesser water and more land mass, or it could be just that at some point this transition had to happen because there was always some land mass available, and the animals which evolved in water, they wanted to probe further and further in search of new predators or new prey, or it could be looking for new mates or new territories. So they had to make the transition, and the transition didn't happen overnight, right? Again, it happened over millions of years, and the first transition that happened is an animal which behaved like an amphibian, okay, which could mostly be in water but partly move into the land. and this animal is called tiktaalik. And this discovery was made very, very recently, how this transition of a fish to an amphibian occurred in evolutionary timescales. Okay, and the amphibian then became more, terrestrial, and then so on and so forth. Okay. All right. Yeah.
Coming back to the movement of a sensory organ.
Yeah.
You said we don't move in the same direction. Is it because it's elevated above our shoulder? Is that the statement?
Yeah, it's more technical. So the question is whether the positioning of the sensory organs in humans is not in the same direction as the movement of the animal. No, it's more the direction. It's a very technical way of defining it. So if you look at the axis along which your sensory organs are present, it's along the top-down axis, right? Like this, right? So your sensory organs are present here. This is the anterior part of the animal, of our body, and the posterior part of the body is your legs, right? So which means that this is the axis along which all your sensory organs are placed. But you are walking in this direction, right? So you can always argue that this is still more or less in the direction of your movement. So I totally agree with that. It's more of a technical way of defining where your organs are placed. All right. All right. So we have now spoken about where your anterior part is. If you have a front, you're going to have a back, right? So the front to back is basically represents one of the axes that I first mentioned in the first slide, right? Our body is organized into a three-dimensional axis, right? And the first axis that I've told you, or I'm telling you right now, is the anterior posterior axis. Anterior is the front of the animal, posterior is the back of the animal. So you can see in any of the animals, right? And most of the organs that are positioned in the posterior part of the animal are involved in digestion, absorption of ingested food, and excretion. Okay, and there's a very simple reason for that. If you eat food and then this food has to be absorbed over long periods of time, the greater the surface area for absorption, the better the absorption will be. So it's natural to have very large structures away from your sensory organs so that you can perform this independent of all your sensory activities. Okay, so that's one of the reasons why sensory organs developed far away from all the other digestive and absorption related organs in the body. Okay, all right. So what I've shown you so far is one axis of the body. Okay, so which is the anterior posterior axis of the body. Okay, in four-legged animals it's horizontal, and in two-legged animals it's vertical. So our anterior posterior axis is top to bottom, right? Okay, all right. Let's talk about the other axis in the body. Okay, so I wanted to mention something else. Okay, so one of the things that is also present in the posterior part of the animal, especially in mammals, is your genital organs, right? So the genital organs have an interesting history. Okay, so did you know that our genital organs are initially specified next to the heart? Okay, so they are not specified. I'll tell you what I mean by specified. Okay, so when they initially form in the embryo, in a little baby, in an embryo, when the baby is inside the mother still, they are actually forming right next to the heart. And the reason for that is because that's how it happens in fish and amphibians and other animals. Okay, so because we evolved from them over millions and millions of years, we have still kept that part of organ formation intact as it happens in fish. Okay, so there are in mammals, when mammals came into evolution, there were advantages of pushing the genital organs out of the body and more towards the posterior, which I will not go into. There are obvious advantages of having your genital organs outside the body. Okay, but the problem is, once the genital organs actually form close to the heart, they actually have to move down to their eventual position, right? And this literally happens in all of us. Okay, when we were babies inside our mothers. Okay, so and the problem with that is that when a genital organ forms close to the heart and it has to move down, it basically has to move through other tissues and out, across the body wall of the, there's a wall here, which basically, sorry, even below the diaphragm. So it's below the intestine. Okay, so it's called a body wall, and it's before the leg that you have, right? So, and the genital organ had to pass through that and come out. Okay, the problem with that is this weakens our body wall, especially in the case of males. Okay, now add on to that we put a lot of weight in a two-legged animal like us. We have a lot of weight falling on our lower part of the body. Okay, plus you have a weakened body wall because the genital organ through embryonic development had to pass through the body wall and had to come out. Okay, and this is one of the major reasons why among males in humans there is a lot of cases of inguinal hernia. So you have heard of hernia? Like hernia is a disorder. I mean, I can't call it a disorder. It's a condition where your intestine is basically sort of like pushing against your body wall and the posterior part of the body. Okay, and this is simply because how we evolved. Okay, simply because why our genital organs are actually passing through our entire body and eventually coming out of the body. Okay, so this is not something which someone should be ashamed of, right? So hernia is a very, very common problem, and this is something which is just a consequence of evolving from pre-existing forms. Okay, all right. So let's talk about the second body axis, which is top to bottom. Okay, now if I just show you a skeleton of a rat like this, you basically have the vertebra, the vertebral column, basically in the top part of the body. Okay, so on the top of the body. All right, sorry. All right, right. So, and that's the vertebral column, and you have the in the other side. So in the bottom part of the body, you have all the soft organs, right? So you have the stomach, the heart, and liver, and so on and so forth. Okay, so it's a very simple organization compared to anterior versus posterior. Okay, so now you're talking about the organization in the other body axis, the top down in a four-legged animal, right? So on one side you have the vertebral column, and on the other side you have all the soft organs. Okay, there is a very simple reason again for why the vertebral column is actually present in the top of the body, because that provides the most stability for the animal. Okay, it can take up more load instead of having the vertebral column in the bottom part of the body. Okay, so the effect of gravity and the load of the entire body on the vertebral column will be much lesser if the vertebral column is positioned on the top of the body. So that's a very simple reason why this organization is seen in four-legged animals. Okay, now this is in the case of vertebrates. What about invertebrates? Invertebrates in the sense like which animals which don't have a vertebral column, right? So I don't know whether it's clear. So it's a bit unclear here. So this is a fly, fruit fly that you will normally see in hovering around your fruits and so on, right? So thank you very much. So in the fruit fly, you will see that in fact this is a heart. This is like a heart and the kidney, right? So this is the dorsal vessel, which is called the heart, and this is the kidney. All the soft organs are actually exposed to the almost to the outside on the top part of the body, right? So how do they then overcome this problem of stabilizing their body plus protecting all these soft organs, right? And one way they do that, we can zoom out. I can do that. Yeah. One way they do that is by having what is known as an exoskeleton. Okay. So in our bodies, we have the vertebral column, which is inside the body, right? In the case of insects, this skeleton is outside the body, and this is called an exoskeleton. So this is a grasshopper, and you can see the skeleton on top of the grasshopper. Okay. And if I look at some closer structure of the exoskeleton, you will see that this is the skin. The what you see in red is the skin of the animal, and above the skin you have this very thick exoskeleton. Okay, the exoskeleton it's an incredible structure, and it has been fine-tuned so much that it's rigid enough, allowing the insects to fly very efficiently, but at the same time, soft enough that it allows the flies to, bump into any obstruction and still not get crushed. Okay, you see mosquitoes in your at your home, and then they keep on bumping against the wall or window or so on, right? Nothing happens to them, right? So this and the reason for that is because of this exoskeleton. It's rigid enough, but at the same time, it's soft enough. So it's this really fine-tuned structure that happened through evolution. Okay, in fact, this structure has been very heavily studied. People from robotics
lost network and you can take a question or something.
Yeah, if you have any questions. Yeah.
The fin to limb concept, is it only for referring it to human cycle, or is there any other living being around it?
So the question is the fin to limb transition is specific to humans? So this is not specific to humans. This is true for any living animal. So you can actually take the fin and remodel it in different ways to give rise to different kinds of limbs that you see in different animals. And we know a lot about how fins form and limbs form in different animals, that you can actually take the part of your DNA that gives rise to the limbs and replace the part of the DNA in the fish that gives rise to the fins with this, and you would get actually limbs in the fish now. So it's that simple in a way, right? So to explain, of course, right?
So,
yeah.
In this diagram, you saying that evolution has come out with the two things that you just spoke about. One is the fins that we're talking about, and just said that you can map the fins evolution to other kinds of fins that are evolution.
Yeah.
And the other thing is about the heart and the face in the fish and then
Yeah.
so that kind of speaks out to establishing the evolution function.
Exactly. So the question is whether evolution, so whether did we evolve directly from fish or not over long periods of time? And the answer is yes. So of course, before fish, there were other primitive organisms, right? So there were organisms which didn't have any vertebra. For example, lamprey, right? Lamprey doesn't have any bones. So they have a soft tissue in their back, which resembles the kind of structure that you will see in a fish, right? And then from lampreys, you got jawless fish. So lampreys are also don't have any jaw. So you got jawless fish. From jawless fish, you got different kinds of vertebrae with fish with vertebrae. And from that, you got amphibians to more terrestrial organisms, and that exploded into having like dinosaurs and so on. And from dinosaurs came birds. Okay. And at some point, all the birds that we see now.
Sundar, I think 25 minutes. You're still at only 2 axes.
Yeah. So how much more time do I have?
So I mean,
15 more minutes?
Easily, 20 maybe, but eventually the chai gets cold. So.
Yeah. I'll be done in 15, 20 minutes. Okay. All right. So now what I've done so far is to basically explain two body axes in all our bodies, right? Anterior, posterior, and dorsal ventral, right? So, and this is sort of the organization that you will see in a four-legged animal and a two-legged animal. So that is the question. Yeah.
So in this I have three axes. So like two bodies, front, back, and right and left.
Yeah. So the question is whether you have three axes or two axes, right? Or you're saying that there are three axes.
I'm asking you if there are three axes.
Yes. So that is the next slide. So we do have three axes, and that is actually the topic of what I want to discuss today. Okay. But then I decided, while I was making the presentation, I realized, oh, actually, it makes more sense to broaden it up and generally discuss about the three body axes rather than focusing specifically on left and right. Okay. But I'll finish with left and right. So that's the plan. Okay. So as someone here rightly said, we have a third axis, obviously, right? So we have a left-right axis. Okay. So that brings to a little question. Can you all point towards your left side? Right? Okay. All right. Cool. Please put your hands down. Oh, that's wonderful, actually. So I've heard a lot about how Mumbai crowd is very knowledgeable, and I can totally see that. Okay. So I'm honest, right? So I've asked the same question in a small place in Germany, and 50% of the people were showing this side versus that side. Okay. So which is amazing to see. Okay. Probably. Yeah. Yeah, that is true. All right. So, I mean, you all pointed towards that side, but my left side is this side, right? So what is happening? So we are pointing on two different directions, right? So how do we understand this, right? Exactly. Exactly. That's exactly the right answer, right? So left-right axis is called a relative axis in colloquial terms. Okay, the way you define what is left and what is right is dependent on how you define anterior, posterior, and front, back. Okay, for me, front, back is in this direction, and for you, the front, back is in this direction. Okay, because our front and back directions are flipped, you automatically flip the left-right axis. Okay, you can think of it as right-hand rule or a left-hand rule, Fleming's rule, which you may have studied in school, right? So this is anterior and this is front and this is left, for example. Now, if I just flip the front side to the other direction, then you can automatically, not that flexible. We can automatically see that the left side is getting flipped to the other side, right? So that's why left-right axis is called a relative axis. Okay, and there is a reason why I started off with anterior, posterior, and dorsal ventral, and then left-right, because that is exactly how within embryos the three axes are defined and established temporally. Okay, when an embryo is growing, it's usually the anterior posterior axis which gets established first, followed by the dorsal ventral axis. And once these two axes are established, automatically you define a third axis. Okay. Yeah.
That's what you want to say in where the evolution starts with the different axes, or the organs are also formed according to that axis.
Okay. So the question is whether evolution, started with a different axis based on what I just said. In a way, it is true. So most of the primitive organisms, for example, jellyfish, or even more primitive organisms, did not have any axis. Remove jellyfish. There are very primitive organisms called sponges. Okay. Now these animals are completely asymmetric. They do not have any defined body axis at all. Then came the jellyfish, which has a single body axis, right? So they're radially symmetric. You can rotate the jellyfish in whatever way you want. They will look very, very similar. So they have a single axis. And once you had a radially symmetric body plan, you transitioned into a bilaterally symmetric body plan, which is basically once you define a second axis, you automatically define a third axis. Okay, because there is only three ways. Once you have two axes, there's only one way you can define a third axis, right? Because these three axes are at an angle of 90 degrees. So you define one axis and the second axis, and the third axis can only be in this direction, right? You cannot define it in any other direction. In a way, it signifies the 3D world we live in, right? All right. So the left-right axis is very peculiar in our body and in most of the animals, because if you look externally, all our organs are paired and symmetric, right? Symmetric meaning they are of the same shape. For example, if I take eyes, they are of the same shape, same size, same position, and in fact, they form at the same time, right? It's true for eyes, nostrils, the jaw, the palate, which is the roof of the mouth, ears. Internally, there are a couple of organs like kidneys, parts of the brain, and so on. Okay, but most of the internal arrangement in the body is left-right asymmetric, right? So heart to the left, liver to the right, and so on. Okay, so how does this distinction happen during embryonic development? Okay, most of what I said today, how the anterior posterior axis is established and dorsal ventral axis and left right, all these are happening directly in an embryo in the first few days of development. Okay, in fact, all of what I said today, the three body axes plus the specification of the different organs in the embryo happen within the first 30 days after a human baby is conceived, which means that even before you know that you have conceived a baby, the entire body plan of the baby and where which organ should go is already determined, right? So what happens later on after 1 month is basically defining a particular shape and size for the different organs, and finally forming the different organs and so on. Okay, all right. So how does this happen? Okay, so let's go to the world of embryos. You have a sperm, you have an egg, they fuse with each other, and you form a single celled embryo. So the field of developmental biology tries to understand how this single cell ultimately gives rise to this complex shape and form, right? So that is the field of developmental biology, right? Now, to understand this, of course, we cannot use human embryos as a model system, right? So we need other models, right? So there are other popular, there are lots of popular models that this research uses: mouse, zebrafish, frog, even plants, worms, flies, and yeast, and so on. So this list is expanding over time. Okay, so we use these as a model systems to understand what is happening within them, but also to understand the kind of organization and the type of functions that each of our organs have. Okay, right. So here is a zebrafish embryo, and this is the yolk of the embryo, and on top of the yolk, you have a bunch of cells that are dividing. Okay, initially you have one cell that divides into 2, that divides into 4, 8, and so on, until a certain point of time when the entire embryo thins and there are dramatic rearrangements of the cells in the embryo, right? And you have an anterior and a posterior of the embryo, and you start forming the different organs. You form the forebrain, the eye, you form all the muscles and the skeleton and so on. Okay, so it's such a beautiful movie that you can, these are transparent embryos, and you can very easily get this kind of movie just using your mobile phone. Okay, so the question is, how do the different organs form in different regions of the embryo, and how are the different axes defined in early embryonic development? Okay, of course, I cannot go into all the details here. I'll give you a couple of important points that is relevant. Okay, one, if you see this embryo developing, you will see that at some point, so they're still dividing, all the cells are dividing. At some point, you will see a sort of rearrangement that is happening. The embryo is thinning, so you can see here. So let me just play that section. Okay, right. So now this is the point when the embryo is thinning, right? So a thick embryo is becoming thinner in this axis, right? And you have some sort of cell movements occurring all over the body, all over the embryonic body, right? So it's covering the yolk, and it's also deciding, where what should go and so on. Okay, and one of the first things that happens during embryogenesis is a process called Gastulation. I won't go into the details, but if I just go through my body, okay, and I open up my body, the first line that I will encounter is the skin, right? And then as I go deeper, you will have muscles or the skeleton, and as I go deeper, you will have all the different organs, right? And this 3-layered organization is irrespective of which part of the body you're looking at, anterior or posterior or dorsal or ventral. You will always have this 3-layered organization. Okay, and that's what is happening during embryonic development, which is the first stage, which involves all these cell rearrangements, leading to formation of 3 layers in the embryo. One is called the ectoderm, which is the outermost layer, and the mesoderm, which is the middle layer, and the endoderm, which is the innermost layer. Okay, so this is something which you need to be aware of. The second thing that happens is what is called as regionalization. Okay, so this is a bit more complex and involved. It involves a lot more background. Before I explain this, so I'll keep it as simple as possible. If you have any doubts, we can obviously discuss. Okay, so now you have the yolk here, and you have the embryo on top of the yolk. Okay, so you can see in this color here, in yellow color or in red color here, we have tagged a particular protein. Okay, so in zebrafish there are 20,000 proteins. Each of your cell has 20,000 proteins, right? One particular protein seems to get higher in concentration right at the margin between the yolk and the embryo. Okay, so what is happening here is that basically the yolk is formed by the mother, right? So the yolk and the oocyte is formed by the mother. While making the oocyte and the yolk, mom actually what she does is put different molecules in different positions that predefines in the embryo what should become the anterior, what should become the posterior, what should become the dorsal, what should become the ventral, and ultimately what will become the left and the right. Okay, so a lot of these things are predefined by the mother even before fertilization happens. Okay, so this kind of activity basically leads to this region becoming the posterior of the embryo, the opposite region becoming the anterior, and similarly you can see that one side getting one type of protein, the other side getting some other protein. So there's a lot of details, but just think of it as something where these axes are predefined while the mother is making the oocyte. Okay, all right. So what happens after this is that, let's say that this protein sort of diffuses through the embryo. Okay, if I add a dye into a liquid glass, into a glass containing liquid, this dye will diffuse from this from the point of where I added, right? So the concentration will be very high in the region where I added the dye, and it will be lesser and lesser in the region where the dye is diffusing later on, right? So basically what happens similarly here is this protein, whatever the protein that the mom has put at a specific place in the oocyte, diffuses all across the embryo, but it forms a gradient. Okay, close to where the protein first forms, you have a very high concentration. Away from where the protein is initially present, you have a very low concentration. Okay, now these cells, because there are a bunch of cells here, the cells are so specialized, they can read this concentration of proteins that are available in its environment and accordingly determine what fate they should enter. Should they enter the fate of becoming a heart? Should they enter the fate of becoming a mouth? And so on and so forth. Okay. For example, in the case of a zebrafish embryo, by about 7 hours post fertilization, you already can map different parts of the embryo, which will become different.
Maybe it might be useful to tell for a zebrafish what is the total time.
Yeah, so I'll do that. Yeah, I was about to say that. Yeah. So basically, what you see here is that at this stage of development, which is 7 hours post fertilization, you can actually map. Okay, these cells will become eye later on in time. These cells will become a heart later on in time, and so on. Okay, so the zebrafish grows for about it grows from embryo to larva to adult, and this process takes place over 3 months. Okay, just the embryonic stage is 5 days. Okay, contrast that to humans, where the embryonic stage is 9 months long, and this process of setting which part of the embryo should become which organ happens within 25 days post conceiving. Okay, so that's what I mentioned before, right? Already before you know that you are having a baby, your entire body plan is determined, including which region of your embryo should become which organ and which cells and so on. Okay, yeah.
Is there anything, suppose we understand genetically that the egg is being fertilized by some certain process. So that means the egg itself understands which cell has created what before fertilization or after fertilization.
So the question is, when does the specification happen, before fertilization or after fertilization, and how does it work, right? So to keep it simple, before fertilization, what the mom does is, to whatever proteins that are present within the unfertilized egg, it tries to segregate some of the proteins to specific locations. Okay, so that establishes broadly this part of the embryo at some point of development will become the anterior of the embryo. This part of the oocyte at some point of development will become the posterior of the embryo, and so on. But of course, all the later on specifications and how many cells should form a heart or a mouth and what kind of shape it should take, those are all completely dependent on the embryonic development. So that is not determined by the mother. Of course, it's partly determined by the mother and the father, because you are bringing those two genes together, and right. So it's the question is whether it's a subconscious process or a conscious process. So the embryo doesn't know what to do. It's something that just happens. Okay, so it's sort of, I mean, I don't know whether to call it a subconscious process, because I can't really define subconscious and conscious when in the case of a, you know.
Maybe I didn't use the right word. I meant it's not a process there. Mother would give a thought to it. Also, I don't know.
Yeah. So the question is whether the mother actually actively gives a thought to it. So as far as we know, no. So it's a completely self-organizing process.
It's an evolutionary process because of the evolution of the different from chimpanzee to human to
Exactly. And basically the same process occurs all through all vertebrates. You can take, because whatever I showed you with the fish embryo, this is exactly the same way a human embryo grows. Of course, the final shapes and forms are very, very different, but the intrinsic molecular processes and the cellular scale processes are very, very similar. But this is a big question here, right? If you have all the like 70% of the molecules which are similar between a fish and a human, right? So given this, how do you actually then form, shapes and forms of, very, very different, which are very, very different in nature, right? So this is something which we don't really understand.
There's a lot of space for research.
Oh yeah, definitely.
I have two questions. Have you identified what proteins are involved in the process of the pregnancy, and have you, like, in the lab, placed those proteins and seen whether those stop the process of differentiation?
Yeah. So the question is, have we identified these proteins in the lab and not? Yeah, we can also take the question later. I can answer this later. Okay. I'll give a quick answer. So we have identified a bunch of proteins which are involved in all these different processes, and we know the identities of these proteins and how they function. The things that we don't know is, of course, we don't have an exhaustive list of all the proteins that are involved in this process. We know some major players, and we know how they act. What we don't know is how they actually set up the different coordinate systems so that you get the three-dimensional axis, and how individual proteins are interacting with each other, giving rise to such an organization. So that is known in some contexts, some it's not known, but we don't really have a full understanding of it. Okay, so I have just two more slides. All right, so let's come into the problem of left versus right. Right. So let's say that here you have an embryo which is about 15 hours post fertilization. Okay. Now, if you can look at this embryo as a tubular structure, right? So it's like sort of like a tube wound around the yolk, right? So if I just remove the yolk and I take the tube and flatten it, right? That's how it will look, right? So you have two eyes, and the brain will be somewhere here. The heart will be towards the left, and you have the muscle and the skeleton forming in a very symmetric manner on the two sides. Okay. Now, if we just go a few hours before this actually happened, that is how it will look, right? So it will have some sort of midline which defines what where the left-right axis is. And let's not talk about how this midline is defined. It's not completely known. In fact, part of our lab will be working on what defines this midline, which tells you where the left and the right should be, and so on. But at this stage of development, both sides of the embryo are equipotential. Okay, the cells on this side of the embryo and the cells on this side of the embryo can form the same structure. Okay, until a critical part in development, when a little organ sort of like turns up magically. Okay, this organ is called the node, which is really present in the posterior part of the embryo. And if I just zoom into this organ, that is how it will look. Okay, so it's nothing but a tennis ball like organ. You can think of it as a tennis ball filled with fluid, and all the cells that are lining the fluid have hair-like appendages pointing into the fluid, just like how you see here. So the hair-like appendages are marked in green here, and these are the cells, and inside you have a fluid. Okay, at a particular point in development, this is what happens. Okay, all these hair-like cells, which are called cilia, they start rotating clockwise. Okay, and this clockwise rotation, you can see that some of the cilia are tilted in some way, right? So they're not directly pointing towards the fluid; they're slightly tilted. Okay, this sort of tilted movement of the cilia preferentially pushes whatever fluid is inside this organ to one side of the embryo. Okay, and this fluid contains some signals which activates some proteins only on the left side of the embryo. Okay, now comes the important point. Whichever cells can respond to this new protein that got activated will become unpaired and form an organ only on one side of the embryo. And whichever cells that don't respond to this new signal that has started from here and reached all the way across the left side will become paired and form paired organs. Okay, so this distinction between which organs should become paired and which organs should become unpaired, in fact, is made earlier, meaning that some cells have the potential to read this new signal that has come up, and some cells that don't have this potential to read the signal. We still don't understand how and when this signaling is basically leading to this differential decision making. Okay, what we know is that this organ is very, very important in biasing one side versus the other. Okay, so in technical terms, this is called a symmetry breaking event. Okay, so the symmetry is broken by pushing some signaling molecules only to one side of the embryo. Okay, so this can be a bit more it can be difficult to understand for some of you. So if you're interested in knowing about it more, we can discuss during chai. Okay, all right. So just my last slide now. So why does it actually move the fluid to the left side? Okay, so if I look at this cilia, which is a hair-like appendage, and look at the base of the cilia. So this is the cell here, and this is a hair-like appendage, which is basically oh yeah, I forgot about that completely. All right, I should move it here. How many people are there online? Okay, I'm anyway in my last slide. Should have done this before.
There are about 10 on Zoom and 18 on
Okay, all right, because they don't see the pointer, right? So anyway, so if you look at this hair-like appendage and you look at the base of this hair-like appendage, that will be the structure of this this hair-like appendage. Okay, and you can see that there are some structures which are oriented in a clockwise manner. They're always turning towards one side, right? So this kind of structure is called a chiral structure. So they only turn on one side. Okay, so they're not turning towards the other side. They're all turning towards. They have a counterclockwise orientation. So right. So this blue structures that you see here, right? So if they have a clockwise orientation, all these structures will turn the other direction, right? So this kind of orientation makes the cilia rotate in a particular way. I'll just that you see here. If you have a different orientation here, you will get a different rotation here. Instead of rotating clockwise, they will rotate the other way. Okay, and this cilia is basically tenths of nanometer in size. The the rotation of the fluid occurs over tens of micrometers. That leads to a symmetry breaking event over hundreds of micrometers. That ultimately leads to an asymmetric positioning of organs in the body. Okay, so in a way, what defines left and what defines right goes right into an understanding of how these molecules are structured within this organ, where you have the cilia rotation. Okay, so people have done experiments where manually people have tried to push the fluid to the other side. Instead of pushing the fluid to one side, you can actually use some air pressure and so on. Within mouse embryos, you can push the fluid to the other side, and this signaling molecule then gets activated on the other side, and you form the heart on the other side. Okay, you form the heart and you have all the organs on the other side. Okay, this is in fact a condition in humans which is called fetus inversus. Okay, where all the organs are completely inverted between the left and the right side. Okay, and you'll be surprised to hear that this condition, which is completely normal. In fact, the oldest woman who lived in Europe had this condition. She lived for 126 years and she was completely normal. Okay, so and she had completely inverted left and right. All her organs that were supposed to be on the left were on the right, and all other organs were on the other side. Okay, so the decision of what goes to left and what goes to right is dependent on the symmetry breaking process, where the symmetry breaking itself is dependent on the structure of the molecules that determine which way the cilia should rotate. Okay, so with that, I will basically conclude by saying that I hope what you would have gotten from this talk is, whenever you see an animal, you don't just see an animal anymore, and you actually try and look at it from its 3D organization, right? So if you look at the dog, you would now understand that it has an anterior posterior axis, a dorsal ventral axis, and a left right axis, which is actually very, very, very similar to how we are, right? So that is how much evolution is conserved. Okay, it's just that the shapes and the forms and the final sizes have changed over time, but the individual organization and how our body plan is actually organized within our bodies remains the same and conserved across a large number of species. With that, I would like to thank TIFR and Chai and Y, and specifically Arnab, for inviting me to discuss this with you.
Okay, I mean, you must admit this is fascinating.
Yeah.
Right? But you should check whether there is any chai left. Okay. What we'll do is, given that there are so many of us here today, I suggest that if you want to have chai, please go and have chai. We will answer some of the online questions and then shut the live stream.
Yeah.
And then we will join you for chai if there's any left. They'll keep some for us, I'm sure.
Yeah.
But otherwise, I suggest that in-person public, you can hang on and listen to these questions. I'm sure there might be common questions as well. But otherwise, you can have some chai if you'd like. Let me start. You said, this evolution, these proteins, you do something, fins, flippers, they change shape. Now that you know all this, can you manipulate this to get any shape you want?
To a certain extent, yes. At least the case of fins and limbs, people have done these experiments where they have tried to make an extra digit, and that is totally possible, right? So because we understand so much more about how fins and limbs and so on form, you can actually go, because we know which part of the DNA actually encodes these different structures. You know which are all the different proteins involved. You know how these proteins come together and act together to give rise to these different structures. Now, which means that now you have all the information which allows you to actually go and edit, leading to different shapes and forms. So one, of course, it becomes an ethical problem at some point. So there is a large amount of discussion as to what can be allowed and not allowed, what is good and what is bad. So this discussion will probably go on for a long time. But technically, for some of the structures, we do have the tools to change the shape and the structure of the shape and form of a particular organ, for example.
Okay, there are some interesting questions on YouTube, and I must thank Siddharth. Siddharth has been patiently typing away answers to many of the simpler questions. These are only the hard ones for you.
Yeah.
Well-trained students. Yeah, what are the interesting ones on YouTube?
Very interesting question from Darshan who asked if the nuclear material provided is completely related to the mother that programs inside, what is the relevance of the father?
Oh, that's a very good question. Yeah. So the question is if the mother actually plays a big role in determining where the different axes should form, what is the role of the father? Okay. So it's an interesting question because the role of the father, I mean, I wouldn't call the role of the father itself, the role of bringing in new genetic material, right? So it's important to have recombination of DNA between the mother and the father, so that you can have new, structures and new forms and new, shapes that can emerge over time. Okay, if you have the same DNA which keeps on, if you do not have any new genetic material coming in, and it's the same genetic material which is keeping on repeating over itself, and let's say that you have a climatic condition where there is a huge change, you wouldn't be in a situation where you can adapt to a changing environment. Okay, the moment you bring in a different kind of DNA, different kind in the sense which has a different sequence, you tend to recombine parts of the mother's genome and parts of the father's genome and so on, which allows you to each individual to be slightly different from each other, allowing each individual to respond slightly differently to changing environments, which is what is very, very important in long time scales.
Isn't that the same thought process behind incest? Because it's the same DNA essentially producing or replicating.
Yeah, exactly. Yeah.
Okay. We'll take all in-person ones. We just finish this and shut the live stream.
Sorry.
So you said that basically you are getting in the DNA. I think the question is the sperm, the content of the sperm, does it have an effect on the placement, the classification of the protein that you were talking about?
Yeah. So okay.
The mother's oocyte that is.
Okay. So maybe I misunderstood the question. So the question is whether the contents brought in by the sperm can actually change the initial body axis that is actually set by the mother. In most of the cases, we don't know of any such situation, except in some primitive organisms. For example, there is a worm which is called C. elegans. Okay, so this is a common worm that you find in decaying fruit bodies like fruits and so on, and this has a different way of doing things. Okay, so the mother doesn't really tell the oocyte where the anterior posterior axis and the dorsal-ventral axis should form. It's rather the sperm which enters, and the position where the sperm enters becomes the future posterior part of the embryo, right? So this is one example, right? So certainly I'm sure that we don't understand everything about what is happening, right? So the sperm also doesn't deposit just the DNA into the oocyte, right? The sperm deposits its centrioles, which gives rise to certain microtubules for biology students, right? And the sperm also brings in certain proteins, which ensures that whatever initial axis that is set by the mother is basically built on. Okay, if the sperm doesn't bring in those factors, the initial axis set by the mother can get lost due to noise in the system and so on.
Okay. Two more questions from YouTube related to, again, evolution and limbs and things like this. Is it possible to grow presently irreplaceable organs, teeth, etc., or damaged organs, etc.? Can you replace them? And a much more thought-provoking question: is there any way to say how evolution would go in future? Like, what would humans look after a million years?
After a million years, okay, all right. So let me tackle the first question. I do not know whether irreplaceable organs can be replaced right now at this point, but there has been a huge advantage, huge improvement in how we deal with cells and how to manipulate them and so on. For example, technology is available in a way where I can take a skin cell from you and I can convert it into a cell that will become a heart, grow it in a petri dish that resembles the heart, and make it functional. Okay, so this has been done not for the heart. I just gave an example. This has been done for the intestine. Okay, so in fact, this provides a huge advantage because now, because everyone is different, and if you have a diseased condition, the drugs that we use will act differently on different individuals, right? So ideally, we will want a situation where we actually test a particular drug in a particular individual before you actually give it to them. Okay, so one way to do that is you actually say you have an intestinal problem, right? You take your own, you can take your intestinal cell, grow it in a petri dish, which is called miniaturized organ, right? So you can grow like hundreds of them, test different concentrations of drugs and different kinds of drugs in each of them, and see how the miniaturized intestine, which is exactly a replica of what is growing, what is within you, how they respond to the different treatments, and go on ultimately defining what should your treatment be, right? So these things are possible, at least in the case of an intestine. But there's a large amount of people who are working on making miniaturized organs for different organs in the body. Okay, and it's just a matter of time. I'm guessing like 4 to 5 decades we will be able to make miniaturized organs of any organ in the body. Okay, so the other question is where would evolution take us going in the future? It's a very tough question. One thing I can say for sure is that if we keep on looking at the phones with a bent neck, we will definitely get a different bone structure here, which allows you to basically look at phones forever, right? It will be an evolutionary adaptation. Right now we get neck pain and so on, but it's just a few generations down the line, this structure will probably get modified and you will no longer have any pain in the body. So that's how evolution works, right? But we will have other problems. So evolution, as I told you, is always a trade-off. If this structure is going to keep your neck bent and allows you to look at phones all the time, this is probably going to lead to some other problems, maybe increased stress, stress in other parts of the back and so on and so forth, right? So, I mean, I can't really tell much more than that, I mean, yeah.
One last question from YouTube.
Yeah.
Is there any related factors in this formation and transformation to fix the lifespan of the organism? Is the lifespan of the organism fixed itself at the embryonic stage?
That's a very interesting question. I never thought about whether it's fixed at the embryonic stage, defined at the embryonic stage. I think lifespan in general is dependent on a lot of different factors, because it's not just about survival of the individual itself, it's survival of all the different cells that make up your own body, right? So ultimately your lifestyle determines how much the individual cells are stressed, right? The more stress you cause to your cells by pollution or unhealthy eating and so on, the faster your cells will decay and the lesser your lifespan will be. So that is my guess. But I never thought about this question whether the lifespan is actually set at early embryonic stages. That's something maybe whoever asks this question, you're welcome to the lab and test it out. Right.
Okay. On that note, online audience, thank you very much for watching. Please keep watching Chai and Why. Sorry you don't get the chai over here. So do come to Prithvi or Referral or wherever it is if you're in Mumbai. But otherwise, watch online. We'll stop the live stream here. In-person public, unstress yourself. Go have some chai. And we'll come back here in 10 minutes. We'll take lot more questions. I'm sure you have lots of questions. Okay.
Yeah. Thank you so much.